You have been learning very interesting unit, that is unit number 8, Physiology of Flowering. Now, in the last lecture or in the previous lecture, you have got idea about the freeze photoperiodism, types of the plant on the basis of a day length required for the initiation of the flowering. So these things you all learn, and at the end of our previous lecture, you have got idea about the difference between short day plant and long day plant. Friends, as we have learned that light and dark period both are important in the initiation of the flowering. So again I would like to say to you, many more factors are going to affect on that one as per the Garner and Allard. Those factors were controlled by the scientists, and only light factor they have changed, and they came to know that light is playing a major role in initiation of the flowering itself. As already I have given you idea, or you might be knowing that the light is received by the specific part of the plant, that is the leaf, and in that leaf the specific substance is produced, and that substance is transported to the shoot or the place where or site where the flowers develop. As we have learned, day length imposes, or day length is responsible for the initiation of the flowering. Similar way, another external or environmental factor is there, that is the temperature. My friends, today we are going to get idea about very interesting physiological process, that is a vernalization. That is a vernalization. So, what is vernalization? Why I have given you idea that external factors which are affecting on the initiation of the flowering. My friends, you have to keep in mind, vernalization actually word is derived from the Latin word that is of the spring. Latin word of the spring. Literally bring into the spring condition. Bring into the spring condition. And also chilling means cold. Treatment ranges from 4 to 3 months. 4 days to 3 months. Site of vernalization, metabolically active meristems of the shoot apex, developing leaves, and sometimes root apex are the site where the vernalization physiological process takes place. Friends, mechanism of vernalization. Here, there are the two theories have been put forth by the scientist in case of giving idea to the specific world related with the vernalization. The theory is hypothesis of a hormonal involvement. One another, that is a hypothesis of a phasic development. These are the two theories which gives idea about the mechanism of a vernalization. First hypothesis, that is of a hormonal involvement. In this one, actually this hypothesis of hormonal involvement is proposed by Chalakian, that is the name of the scientist. And according to him, vernalin is produced in some biennial and perennials at low temperature in a long day plant. Anthocyanin is converted into a GA. As anthocyanin is present in the long day plant, the plant which flowers after receiving a long day condition for the emission of a flowering. Anthracene plus vanillin causes flowering in a long day plant, but in short day condition, vanillin is not converted to the chew because there is a lack of anthracene. Hence, flowering does not occur. Now, second hypothesis, that is of a phasic development hypothesis. It was proposed by Lysenko. The annual plants seeds have a series of development which occurs in the predetermined sequence. Next is the phase only initiates the termination of a previous phase. There are actually two phases, that is thermophase is there and photophase. In case of thermophases, the vegetative phase, which requires low temperature 0 to 14 degrees centigrade and suitable moisture and aeration condition. This stage is a variable length and it depends upon the nature of the plant. So we know that for the initiation of a flowering during the vernalization, plant will have to go through the two phases. First phase, that is a thermostage phase, and another, that is a photophase. So in case of thermostage, the temperature required, that is between the 0 to 14 degrees centigrade. Along with that one, suitable moisture and aeration is also required there. And that thermostage is a variable length and it depends upon the nature of the plant. So that is different to different plant. In case of the photophase, it requires a high temperature and depending on the light share, the vanillin helps in synthesis of a florigine, which is a stimulus of a flowering. Thermophase should be followed by photophase. You have to keep in mind, my friends, in case of second hypothesis, that is the phasic development hypothesis, always there is a two phases are there, thermostage or thermophase is there and photophase is there. So always this thermophase is followed by photophase for the development of a florigine, isn't it? Now, after getting idea about the vernalization, you have to keep in mind devernalization is also there, isn't it? So what is devernalization? Reversion of a vernalization by high temperature treatment. We know that in case of the vernalization, there is a requirement of a chilling treatment, cold temperature treatment for the initiation of a flowering in the flowering plant. But in case of the devernalization, that reversion of a vernalization by high temperature treatment is known as a devernalization. So whatever vernalized effect is there, that effect is nullified or reversed, or reversion takes place by the treating that plant with the help of a high temperature. It is achieved by treating the vernalized seeds or buds with high temperature. It should be done immediately after the vernalization. Sometimes low irradiation also brings about the vernalization. You'll have to keep in mind. Immediate. Suppose vernalized seeds or buds are in your hand. Immediately you will have to treat that seeds or bud with the high temperature. Otherwise devernalization will not take place. Okay. Now, what are the applications of a vernalization? Uses, significance of vernalization. It helps in shortening the juvenile or vegetative period of the plant and brings early flowering in many plants. This is a saving the time to complete the life cycle. My friends, we know that most important phase in the life or physiological process in the life of the flowering plant is a initiation of a flowering and then fruiting yield that is there. We want always each and everything get shortened. Means vegetative growth it takes suppose the 2 years and then flowering is there, but we are not interested in case of waiting for the 2 years. Within 1 year we want that flower should be flowered or should develop the flower, and at such a conditions the vernalization treatment helps us to make that plant to produce the flower within a 12 month, not after the 24 month, isn't it? So that is the most important application. Early flowering induces early formation of seeds and leads to the early harvest, isn't it? Within a 1 year, flowering, fruiting, yield is there, isn't it? So that we can induce early formation of seeds. It is useful not only to the temperate but also to some tropical plants like wheat, rice, millet, and cotton, isn't it? So harvesting period we are going to make early or shorten the, we have to say that, vegetative growth period. Increases yield and resistance to the cold and disease causing fungi. Friends, as we know that, suppose specific period is that flowering to the plant is in a month of a May is there, isn't it? And some fungi, some diseases, some, we have to say that, factors, adverse factors which are causing damage to the yield of that plant in the summer, or we have to say that in the May. Suppose 2-3 months early harvesting took place in February or in March, we have to say that. So definitely the disease causing fungi will not get a specific hot atmosphere or specific period for their infection to the specific plant, because already or early the plant has been removed from us, isn't it, or harvested from us. So that is, again we know that resistance to the cold we can increase with the help of a vernalization. The kernel wrinkles problem in triticales can be solved by the vernalization process. Also, vernalization is useful in case of crop improvement. So these are the, we have to say that, applications of a vernalizations. Now friends, we have learned very most important two processes which are taking place in the life of the plant, that is a photoperiodism and vernalization. Now, then what is the difference between these two that we have to learn right now? Here my friends, you have to keep in mind, photoperiodism, that is, it is a response of a relative length of light and dark period, that is photoperiodism. But here, acceleration of the ability of a flower by chilling treatment, isn't it? Acceleration of a ability to flower by chilling treatment, that is nothing but the vernalization. Photoperiodism, in case of that one, stimulus is pursued by mature leaves. As already I have said to you, florigine synthesis takes place in the mature leaves, and that is transported to the site where the flowering takes place. But in case of vernalization, cold temperature treatment stimulus is pursued by the active meristems, maybe the shoot apex, root apex, or we have to say that the leaf. That is the difference between the photoperiodism and vernalization. Photoperiodism not only prepares the plant for the flowering but also intimates the flowering. That is most important. Itself does not induces flowering but prepare the plant for the flowering, that is vernalization. Photoperiodism or initiation of a flowering is mediated through the florigine, but vernalization, that is mediated through the vernalin. The effect of photoperiods cannot be reversed. Keep in mind, once the photoperiod is given, initiation of a flowering is possible. That we cannot reverse. There is no reverse, isn't it? But in case of vernalization, already we have seen the immediately, suppose the vernalized seeds or birds again treated with the immediately high temperature, then reverse is possible in case of vernalization, but that is not possible in case of photoperiodism. Photoperiodic can be transmitted from the one plant to another plant. Suppose one plant is treated with specific photoperiod. I will give you example. That is, we know that we are having the two plants in our hand, A plant and B plant. A plant is a long day plant. B plant is also long day plant. But A plant we have kept in a short day condition, and B plant we have kept in a long day condition. B plant will flower there because that is naturally in the long day condition. But suppose some part of that one is grafted on the short day condition, but plant having a capacity to flower in a long day condition, then the plant, although present in a short day condition, but that is grafted with the plant part which is taken from the long day condition, then also flowering is possible, isn't it? But vernalization cannot be transmitted from one plant to another plant by grafting, except one of the examples there of henbali. Now, photoperiodic stimulus is interrupted by the dark period. Again, vernalization stimulus must be applied continuously. Uninterrupted is the no flowering could be possible in the plant in case of, although that is a vernalized one. Same case in case of the photoperiod. Here the uninterrupted photoperiodic stimulus is uninterrupted by dark. So this is the difference between, we have to say that, vernalization and photoperiodism, that you have to learn, you will have to write, you will have to concentrate on each and every word what that is going to tell to you, and then only it is possible to understand the difference between the photoperiodism and vernalization carefully. Friends, as you know that this plant physiology paper is although very simple to understand, but at the time of writing all these things answer, then it would be a very difficult. Or if you found that it is a very simple in case of understanding, it could be difficult in case of writing the answer, or it could be a simple. Oh, you'll have to keep in mind all these things very well. You'll have to study, you'll have to observe again and again. I will say to you, you are the science faculty students, then you'll have to have a keen observations, you'll have to always look about you, you'll have to see some plants, go approach the plants, then see the nature, structure, the changes taking place into the plant often and often, and that will help you to make your concept clear first of all, and then it is possible. Why we have to go to improve the plants, isn't it? In case of increase the production, increasing the demand of a basic needs by the human being, and for that one we'll have to increase the product, enhance the production of each and every things. That is in case of the plant yield, and that is possible only by going to learn some physiological processes, and we'll have to turn that physiological processes as per our need, or as per, we have to say that, our willingness. So that we have to learn here very well. So my friends, with this I said to you, my friends, once again, you'll have to prepare notes on yourself. Then only it is possible to you to understand each and every concept, isn't it? And another thing I will say to you that, my friends, as we have learned actually 8 topics, one topic we have to learn again, that is of the 7 topic unit, we have to say that, not the topic unit, that also we will learn related with that of growth hormones is there, isn't it? So my friends, as I have said to you, you will have to read, you will have to go through the textbooks, reference books, Again, the material, PPT, in case of these things, are available on YouTube of other authors or other teachers. That also you'll have to hear and make the concept clear of each and everything, isn't it? With this note, I say thanks to you. See you in the next lecture.